All right. Hello, everyone. Back for another little show. Hopefully the bit rate isn't going to be too bad today. We'll see how things kind of go. My kids are still trying to watch YouTube upstairs, so that makes it a little bit more difficult. So today we're going to be talking about stats. I see there's only a few of you guys joining us today. You guys get a personalized lesson about statistics and what's going on, what it means, and how to improve your reports. So it shouldn't be too long of a class. I only have a few slides to show you to try and get you a little bit up to speed to what is going on in the lab, because I have a feeling that some of you didn't understand what the statistics were actually looking at and what we were trying to compare. So I'll chat a little bit about that. So this is your opportunity to ask any questions that you need to ask so that you can improve your report. So remember, I'm on Discord again if you guys want to chat. I think Sue may be joining us as well. So if she is, let's try and keep the chats in YouTube live today. And I'll check on the screen every once in a while, and I'll take your comments that way. So hopefully you guys can hear me okay and everything's fine. So let's just get into it a little bit and we'll see what you guys want to know. So what are statistics really used for, right? They're trying to help us understand whether there are differences between groups, whether that's between populations, between species, between areas, whatever that is. When you're comparing things to see if there's a difference between them. So one of those things may be, let's say this blue bar right here, and this bar has a mean value of this. Now this could be height, this could be weight, this could be growth rate like in our experiments, this could be color, it could be anything, any trait. And then what we're using statistics, and what we want to know first off is, does this on average, this mean value on average differ from this mean value? So now we look at these two things visually and we say, well, they look really different, but we can't say anything more without actually using any kind of statistics to determine whether they are really different. So that's generally why stats are used. They're allowing us to compare different things to see if they truly are different. There goes an ambulance. Hopefully that doesn't get picked up. We'll see. Okay, so I'll keep going. This looks like there's a statistical difference. This one less so. So again, we need those stats to be able to determine whether that really is the case. Okay? So in our experiment, what I want you guys to answer is, what was the question that we were actually exploring in the fungal prac? Like, you guys went through all that work, you inoculated all those plates with a whole bunch of different species, but what were we trying to understand by doing this experiment? And this is what I want you guys to be able to tell me, and I want to see if you guys do understand what the purpose of that experiment was. So I actually forgot to get myself some tea before I started the show, so I'm going to let you guys think about that question, and I'm going to fill up my cup with a little bit of hot water, and I will be right back. So let's see if you guys can answer that question for me. What was the purpose of the fungal prac? All right. So no one's jumping in on anything today, so it looks like we don't have the people who are normally... Hey, there we go. Thank you, Ro. So to see if competition has an effect on growth rate. Yes, that's exactly it. We wanted to know whether individuals, when they had to compete against one another, whether that affected how quickly they could grow. So what we did was we had two different species, right? Two different species of fungi, and we had to determine if competition was affecting growth, or when species 1 was competing against species 2, we had to have a bunch of other different treatments, didn't we? Because if we have species 1 and species 2 on a plate, that doesn't really tell us anything. It tells us how well those species do just together, right? But to be able to compare them to different groups, we needed those other treatments. And let's see if you guys remember what those treatments are. Have a think, but I won't wait for you this time. We'll just get into it. And one of those is, how does species 1 do against, species 1 do against itself? And how does species 2 do against itself? Then, of course, we needed 2 other treatments kind of as controls, which was how does species 1 do on its own, and how does species 2 do on its own? And that's why we had 5 treatments. That's also we could kind of compare how species 1 and species 2 growth rates did when they competed against each other. So does that make sense? So hopefully it does. And the next thing I'll ask is, how did we calculate growth rates? Right? We took photos at 2 different time points, right? And we wanted to see how much they grew from time 0 to time point 1, and from time point 1 to time point 2. So those are our growth rates, but we only ended up using the second one within our analysis, right? So hopefully this all makes sense to everyone so far. If it doesn't, let me know, and I'll repeat anything, because these are the fundamental things you guys have to understand. Before we do any other statistics, we have to understand why we're doing the experiment and what exactly we're looking for. Because the stats themselves only allow us to determine whether our predictions or our hypotheses were correct. And to understand whether our hypotheses are correct, we have to understand why we were doing the thing we were doing, right? And this is what I get from some of the reports that we saw. Some people went through all the motions, but they didn't understand what we were doing, and as a consequence had a lot of trouble understanding what the stats meant. Okay, so when looking at the data, Ketty, I feel somewhere in millimeters, another were in meters, so we could really, could we properly compare? I think that was just a typing error. They're all probably they were all in the same, the same units, because otherwise, yes, you wouldn't be able to compare. But also, we're going to assume that they are in the same units. And this is one of the problems, of course, of having a huge class collect data together, because different people do things differently. Some people may have made some mistakes accidentally, which is what a mistake is. So, the more people that are involved, the more difficult, obviously, gathering class data is. But that's just, how things kind of go. And we do the best that we can. So I'm not really concerned with potential errors in the data or things like that. What I want you guys, what we really wanted to know is, how are you using the information that you've gathered to make conclusions about understanding the system that we're exploring? Mistakes happen everywhere, and you can't avoid them sometimes. You can minimize them, and in a class of 50 or 60, that's hard. But so that's why we aren't going to include the fact that there might be some errors in the data themselves. So why did we use the second one? I used both. Okay. Even better if you did. Some people only used one. Some people used the earliest and the last, and this is part of the problem. So I'll get to yours in a second, Carrot Destroyer. So using both is even better, because then you can take a look at how early growth and late growth is affected by competition, because we would have a hypothesis there, right? Early on, when individuals are growing, there's a lot of space, and individuals can move out relatively quickly. But at some point, they're going to meet up, and when they meet up, that's when competition starts. And that competition really is going to start when there's less area. So we could expect that the growth rates could actually differ between the 2 time points, right? Because the second time is likely when more competition is actually happening. So yes, you can talk about potential mistakes in discussion, but I don't want that to be a focus of the discussion, okay? I want you to, so I know in early undergraduate and even in high school, what you're taught is, well, you've got to really nail explaining why those mistakes, what could have affected everything. Those mistakes could have really, affected how you interpret those results and your results. Yes, that's true. But like I said, mistakes are unavoidable. You're trying to minimize those mistakes within the experiment that you're doing. So what I want you to think about when you're writing your discussion is, these are the data that I have collected, or our class has collected. This is helping us understand the system that we're exploring. I will use these data to best explain what is going on and interpret these data to better understand what's going on between the two species. You can talk about limitations, but limitations are more of we designed a study this way, and as a consequence, we couldn't explore these other questions because that would require a different statistical design. So that's a limitation of our study, for example. But errors and problems with the data, I don't want you to focus much on that or any on that, because that's implying that you're not good scientists and you've made mistakes, which, you're practicing to become better scientists. So I don't want you thinking about the mistakes that you've made in the experiment that would lead to you not seeing the results that you thought. What I want you guys to do is, these are my results. I'm interpreting these results because these are the results that I received. And they are right because we did the experiment together and this is how they turned out. So does that kind of make sense? And I know it's a little bit of a different mindset than what's pumped into you during first year and especially in high school. But let's not think about mistakes as you guys making mistakes or errors within your study. Everyone does, but you do the best that you can with the data that you get. Does that make sense? So if there's any other questions, keep throwing them in. So how do we calculate growth rate? I think I answered this, but I'll say it once really quickly again, is we subtract how big you are from the different time periods that the photos were taken, and you divide it by the days that it took to get that size. And then you have an average growth rate per day, right? So I'll answer your second question near the end of this lecture as we go along. So here we have, let's say this is species 1 growing on its own in blue, species 2 growing on its own in red, okay? Like I said, we have more, actually, you know what? I'm going to jump back because I forgot to say, no, I don't want this to happen right now. What is the analysis that we would use to explore whether the means are different when we're comparing two groups? Can anybody remember what that test is called? Because I want you guys to get a little familiar with the statistics and the tests and the names of these things so you don't feel so afraid of them. Because all statistics really use is all it is really a little bit of maths to help us understand the logic that we're trying to use to explain a system. So does anybody remember what this test is actually called when you're comparing two means from two different groups? A continuous variable, so these are the means, a continuous variable, let's say height in population 1 and height in population 2, and where you have your two categories are these two different populations. And it's a t-test, exactly. So it's that simple. You're just comparing two means from two different groups. And what we're doing now here is we have two means from two different groups so far, which is species 1 by itself and species 2 by itself. But then what we add is species 1 competing against itself and species 2 competing against itself. Then we have the most important one of all, which is species 1 competing against species 2. Right? So now we have 5 groups. Because we have 5 groups, it's not a 2-sample t-test anymore. It's just something with multiple groups. But you're kind of doing the same exact test. Okay? So it's not really any different. There's just 5 categories. and means for those 5 categories. All right? I don't know why my Discord doesn't like updating. There it is. And yes, no, Lulu, I'm just trying to explain the stats. So no, you weren't. Well, I'll get to answering how you're supposed to display the stats in a little bit. Okay. So hopefully that makes sense. So this really isn't any more complicated than a t-test, even though the script looks like it was. But the other thing that we have to add in the statistics is our random variable. And that's your group ID. That was each of your individual groups. Because we added that because there's likely going to be some variation in how the different groups perform the experiment. So we want to take that into consideration into our stats. All right? So that's why we have our stats as, is growth rate affected by treatment with these 5 different treatments in there, with group ID as a random factor because we understand that different groups may have done things differently, so we have to take that into consideration into the statistics. All right? Oops, I just turned it off. So, Chapo, exactly. This really is an ANOVA. Okay? An ANOVA is a t-test, but with multiple groups or multiple categories. So this is called a general linear model only because we're trying to control for random variation between groups and how groups did the experiment and calculated their growth rates. See, so hopefully this is all starting to come together and the statistics themselves are making a little bit more sense. Okay? Statistics seem really, really complicated, and they always are until you really dive into something. This is why we kind of threw you into it, so you can kind of think a little bit about what that experiment is and what you're doing and what those comparisons are. But when you really break it down and you think about it, it's like all I'm doing is looking at how these different groups perform against relative to one another, and I'm taking into consideration that different groups did things differently. All right? Katie, I'm not sure what you mean by your comment, so if you can just clarify that a little bit. So hopefully that kind of makes sense. So I'm going to go into what the output looks like. So remember, we had 5 groups, right? Species 1 on its own, species 2 on its own, species 1 competing against itself, species 2 competing against itself, and species 1 against species 2. So then we had this, and you guys probably looked at this and were like, oh my God, I have absolutely no idea what this means. Why are these guys making us do this? But some of you got it, which is great. Some of you didn't, but that's okay. You get another chance to do it now, which is why we're doing it this way, so you guys get a chance to improve. It's the only way you're going to learn. Okay, so what we have here are our different treatments, right? Species 1 versus species 1, species 1 versus species 2, species 2 alone, species 2 versus species 2. And you notice we're missing one, right? That's even though we have the mean growth rates from the different treatments. So that's probably confused you guys. So what is missing there? Let's see. Right. Okay. I'll explain that in a second, Ketty. I understand what you mean now. So I'll talk about that near the end again, and remind me if you don't hear it. But I'll help explain what the difference between a results and a discussion section is, and when you would use t-values and the stats and mean growth rates and things like that. So what's missing here? Someone's typing it in. Hopefully they get it right. But I won't wait too long. Yes. Okay, you've nailed it. Okay. So species 1, the estimate size for treatment 1 is missing. And that's because the way statistics actually work and how what this model is actually doing is it's saying this is the estimate of growth rate of species 1 versus itself against species 1 alone. This is the estimate of growth rate of species 1 against species 2 compared to species 1 alone. And this is compared to species 1 alone. And this to species 1 alone. So species 1, the estimate for species 1 here doesn't exist because what the stats is doing is it's comparing each one of these groups against the growth rate of species 1. All right? So that's why we only have 4 comparisons here. So hopefully that makes sense. Now you can hopefully start seeing how the stats work, what the statistics are really doing, and to some extent, how you could extend the statistics to look at very particular kinds of things. So here, what this is now saying is this is an estimate, and this is essentially how strong of a difference or how strong of an effect this treatment had on growth rates relative to species 1. This is your degrees of freedom, which is essentially your sample size. And then this is your t-value, which is essentially exactly what you would get from a t-test. And this t-value allows you to get an estimate of a probability, the likelihood of a difference between those two treatments. In this case, species 1 versus itself versus species alone. And what we see here is there is no significant difference between their growth rates. So let's take a look really quickly at the mean growth rates of species 1 alone versus species 1 against itself. And what we have here is the growth rate is 188 millimeters squared per day. And then here's 174. Let's not get stuck on the units now. And you notice they're not very different, right? So here, again, now we have the comparison for species 1, the growth rates, the average growth rates in here compared to species 1 alone. Not quite significant either. Species 2 alone is very different, and species 2 versus itself is very different. So now if we take a look here, what we've got again is species 2 growth rate is, I'll use my other hand, is much higher than the species 1 growth rates, right? So does that make sense how the statistics themselves are supporting your observations? That's what they're doing, right? The stats themselves don't say anything without this information. So, Ketty, hopefully it's starting to make a little bit of sense about when you would use your t-value and your growth rates. So what you're saying here is species 2 grows on average faster than species 1 on its own. And we know that because they are statistically different from one another. So you state your growth rate and you state your statistics as support for making the statement that they are statistically different growth rates. Okay? So I'll say that one more time, and if you guys have any questions, ask. Your growth rates are here is what you've observed. Your stats are used to support whether your observations are different from one another or not. So in your results, you would say whether they are different or not. You would compare the means that you're comparing and say what is different or what is not different from one another, and you add your statistics, your t-value and your p-value in brackets after that. So when you go, what I want you guys to do when you're writing this other one, this new version of this manuscript, when you're looking at those other manuscripts that you're referring to, that you're referencing in your paper, take a look at how they talk about statistics. And hopefully what I've said here is made a little bit more sense of how they explain the statistics and what they state in the results. So in your results, you will be stating, yes, they're different, or no, they're different. We compared these things and we don't see differences between them, or we do see differences between them. Here are stats to support this. And then in your discussion, you explain why those differences exist. You discuss what those differences mean. You discuss why you saw differences, what may lead to those differences, what those differences mean in a biological or competitive sense. All right? So your results talk about what the results of your experiments were with statistical support, and your discussion talks about what those results mean in a biological context. So hopefully that's cleared things up and cleared when you use t-values, when you use p-values, when you use mean growth rates, when you talk about what, where in a paper. All right? So, of course, for some reason, Discord, give it a second, it's updating, here it is. Okay. How do we discuss the table in our results section in terms of the actual growth rate and treatments? So hopefully, Alexa, I've answered that question. That kind of makes sense now. If it doesn't, ask me again. But hopefully that's a little bit clearer now. What does the p-value for SP1 by itself mean? There isn't one, right? So there isn't a p-value for SP1 because you can't compare that treatment to itself, right? These p-values only exist because the means are being compared to SP1. So does that make sense, Kiki? Ask me again if it doesn't, okay? And is a high SD in the second table a bad thing? You mean in here? No, it's not a bad thing. There's no such thing as a bad thing in this kind of sense. What it means is this was the mean and there was a lot of variation, right? So if this was the mean, some groups SP1 grew, on average, everyone's group SP1 grew 188, but some groups grew more than that, which was, let's say, almost 400, because if you add the mean and the standard deviation, right? So what that standard deviation means, when you have a high standard deviation like that, it just means there's a lot of variance within that group, right? And yes, it does. It could mean that there's variation between different lab groups, and that's where our random variable comes into play. Because the random variable itself, if that random variable can explain a lot of the variation in the results, if that's significant, then that means there's a lot of variation between groups. If that is not significant, what that means is that there's just a lot of variation in the growth of SP1 on its own. And we don't know what that is because we didn't test for that within our model and our experiment. Okay? Would you repeat the test using SP2 to determine whether anything is statistically similar to it? So Chapo, now you're starting to extend these statistics. This is what I was talking about when you want, how you can take this further. So we can actually change the reference species. So rather than making it SP1, we could make it SP2. Or we could make it SP1 versus SP1. Or we could make it SP2 versus SP2. But we wouldn't make it SP2 because that's the one we're most interested in. So you can actually go back and change the reference in a statistical model to change what these different treatments are being compared to. And you will likely get different statistical results to some extent, depending, and you could probably guess which ones. If you change it to SP1 versus SP1, you probably won't get very big differences because you have very similar growth rates here, right? But if you change it to SP2, you probably will. Then what we probably wouldn't have is these two wouldn't be significant because you're comparing the growth rates to SP2. And hopefully that's not too confusing, but what it means is you're changing your reference, and as a result, you will change your statistics. So you can go and do that, but there's no need to do that in this report. Okay? I found the effect of different treatments within each group, then did the statistics on those slightly more processed values. Yes, that's fine. I'm happy. I can take a quick peek at your results, Ben, and I can take a quick look. So you can message me directly through Discord. And good. I'm glad that nailed it, Alexa. Thank you. Are you able to say what figures should be included in the results tables and graphs? Now, what we can do is, I don't want to tell you, okay? Because I don't want to tell you what you should be doing, because to some extent, you guys should be able to figure that on your own. So, but we can talk about it. So if you guys want to start throwing in some comments in there, we can have a little bit of a discussion of what you think is important and what figures or tables should be there. But I don't want to tell you what that is, because me telling you that isn't going to help you learn this at all. It's just going to end up you doing it and then you getting to your next lab going, oh man, I wonder which figures and tables I should end up using in my report. What I want you to do is be able to understand why you're selecting the ones you are in this report, so you can do it again in the future. Our goal here is to help you get better at this kind of stuff. So, I'll let some of those comments kind of pop up in the discussion. I'll go down another question, and then we'll jump back into that idea. So should we construct a whole table in the results of the statistical test, or just state it in the results? You can do either, okay? But what a table does, a table like this would provide you with all the stats. Now you can put this in a written format, and that is totally fine, which means you don't need a table at all. But that means you have to give us, in written format, all these results. Does that make sense? So that means you have to explain all these comparisons. All right? So either one is fine. And even if you do add this table, you kind of want to explain the differences within the results. Here we do see a difference, here we don't, and we have support for that. Look at table 1. All right? So, effective different. Okay, I'll jump back to you for a second, Ben. Why are we more interested in species 2? We're not interested in most in species 2. We're most interested in species 1 versus species 2, right? Because we want to know how these two different species do when they compete against each other relative to when they don't compete directly against one another. So we're interested in competition, right? So we're interested in how organisms do when they have to compete with one another. So we're using species 1 as a reference because species 1 isn't competing against itself when it's by itself. So we compare species 1 versus species 1 against itself, and we see how it does in that situation. That's a valid comparison. And then we do the same thing for the other treatments. So this one is just comparing how species 2 grows on its own versus species 1. And you see there's a big difference. So hopefully that makes sense. So where is the random variable in our stats? It should be output near the bottom where it says random variable. I didn't copy it into this PowerPoint presentation. So let's see if we can take a look at it later. I'll take a look at the output. And we can see, I'll see if I can send a screenshot of it here in Discord. Yeah, don't present raw data, don't present the same data twice. Present the data that answers the hypothesis question. Absolutely, okay? So Lulu, box plot? Could definitely use a box plot. Box plots are really, really nice. So what's the difference between a box plot and a bar graph, for example, folks? And for example, what is the difference in the information that is provided in a box plot versus a bar graph? Why don't you guys tell me that? And I will say that even the scientific community in and of itself has used bar graphs for a really, really long time, and sometimes they're really, really suitable, but even we're moving away from that because bar graphs often don't provide a lot of information in a single glance, while box plots are often better. But I want you guys to tell me what does a box plot show us relative to a bar graph? Yes, that's right. A box plot compares means with error bars, and it has even more than that, doesn't it? Because it has those error bars, but it has something else. Because bar graphs can have error bars as well. But bar graphs tell you a mean with a standard error. Well, box plot tells you a mean with error bars, but what else is in there? What does that box signify? See if you guys know. It does show more of the variation, that's true, Lulu. So it does allow us to see more of the variation, but it provides specific information, right? In that box, you have your horizontal line, which is your mean. Then that box represents the first standard deviation, where the vast majority of individuals lie within. Then it has those error bars, which can show you, for example, up to 95%, where the other standard deviations sit. And then you have often you have little points that sit above those standard error bars. And those points tell you outliers. So you can see in a really quick glance, you can see a lot of information when comparing box plots compared to a bar graph, for example. Now you can go even further because there's something now called the Violin Plot, which is very similar to a box plot, except it's essentially a frequency distribution. So the Violin Plot looks like a violin, and it tells you very, very specifically where most of the individuals lie within a plot. So that's kind of really cool, and that's a new way that scientists are starting to show variation within a group that you're comparing. Yes, exactly. So the interquartile range. That's another way of saying all those standard deviations. The standard deviations and your error bars. Excellent. So good. So hopefully that makes sense to everyone. I actually don't have any more to talk about. We'll go back to the beginning because it's just a nice picture. So if you have any other questions, ask away here. Yes, we're gonna go as far and assuming normal distributions for this kind of stuff. I'm not gonna ask you guys to go that far to start guessing, looking at distributions and making those. That's a bit more complex statistically. And listen, it's fine. That's not that important right now. Yeah. So I'll end it there unless folks have any more questions at all. I'll just wait here and finish my tea if you have any questions. And tomorrow, all right, should we, I don't know why there we go. Should we cite other studies and other experiments in discussion? Yes, you do have to. Because what you're doing is you're comparing your results to other individuals, or you're making inferences of your results in discussion, explanations of why the species are competing in this kind of way, what competition does to individual performance, and you want to support the inferences you're making from other people's experimental results. And that's where you'll be finding other data to support the conclusions you're making from your results. So absolutely, you do need to find other papers that will support the conclusions that you are making in the discussion. And sometimes you will find papers that come to different conclusions than you have. And that is also okay, and it is fine for you to use those as reference material for your paper. Then you have to explain why you think those differences exist. Okay? So you would present the t-values and the p-values in the tables, or you can do it alongside the means within the text. Either way is fine, as long as you provide us with the p and t values for that. And you should somewhere tell us the means whether that's in a figure or whether that's in actual raw values, whatever that may be. So how would I recommend finding more papers to reference in our reports for our intros and discussions? So the best way is if you go to Google Scholar, you can start Googling and looking for some papers that have explored competition, and that could be in any kind of organism. It doesn't necessarily mean it has to be in the species of fungi that you have been exploring, right? So you can look at any organism, any competition, but you want to make sure it's relevant to the type of competition that you are exploring in here. And here is interspecies competition, right? How two different species compete for access to resources specifically through growth rate, all right? So if you start putting those search terms into Google Scholar, things like growth rate, interspecies competition, fungal competition, plant competition, animal may be a little bit too far out there and too different for you guys at this point. So if you start putting those search terms in there, you'll get a ton of different papers, and you'll just take a look at some of the titles and do your first scan and see which ones may be relevant. Then you highlight a few that are relevant. You might look at their abstract to see if they are right, and if they are, you read some of those papers and you see exactly what they've done. You may find only one or two really good papers. There are probably a ton more, but you may find one or two that you're really happy with, and then what you can do is you can look within the references of those papers to see what those people cited, all right? Excuse me. And then what you can do in Google Scholar is search for that paper and see which other papers have cited that one that you're holding in your hand. All right? So you can search by using search terms. You can search within a paper to see who's cited, what else they've cited, and you can see what other papers have cited the paper that you found. So those are the three main ways to find relevant literature. All right? And so there's heaps of stuff out there. You will not find a problem or have a problem finding any of this kind of stuff. You just have to look for it. And those are the best ways to find those papers that you're going to need. So hopefully that explains. So that's the end of the questions. Yeah, hopefully that explains everything. I'll sit here for a little bit longer and you guys can watch me drink tea. And then, yeah, then it'll get to a close. So when referencing specific ideas, if the paper you're looking at cited that idea from another paper, which paper do you reference that idea from? If you're looking for the original author on that, okay? You can't cite a paper about a specific idea if that idea wasn't explored in that paper. If they're citing a different paper for that, then you have to cite that earlier paper. So you're going to have to read that earlier paper. And it's good for you to read that paper because you might find other literature citations in there that might be relevant for you in your report itself. Okay? So you have to go back to the primary source when you're referencing a piece of material. All right, I think I'll leave it there. And because I got in trouble for not rocking out at the end, we'll have a little reggae to bring us out. So if you guys have any other questions, you can throw some questions in the Discord. I can answer things as we go along later on. Please do take this seriously because we don't have an exam. So this is a big chunk of your final mark because it's your final big report. You've gotten a lot of feedback. So take that feedback and improve. We really want to see all of you guys do really well on this, okay? Because it's going to help you guys get better for any kind of future writing that you're doing. All right, so I will leave it there and have a great day, everyone. Enjoy physically distancing, and hopefully you guys haven't watched everything on Netflix yet. So chat soon. All the best, guys.